For decades, doctors viewed head and neck squamous cell carcinoma, or HNSCC, as a predictable disease. It was almost exclusively diagnosed in older patients with a decades-long history of heavy smoking and drinking. As global smoking rates dropped over the last few decades, the logical expectation was that cases of this specific cancer would drop right alongside them. Instead, rates of cancer arising in the oropharynx, the back of the throat, soared. These cases appeared in a completely different demographic, younger men with zero history of using tobacco. The explanation for this spike is that HNSCC is not a single condition. It is two distinct biological entities, driven by entirely different causes. There is the classic, environmentally driven cancer, now classified as HPV-negative, and a virally driven cancer, classified as HPV-positive. Treating head and neck cancers as a single, uniform disease for so long obscured a completely new, virally driven epidemic hiding in plain sight. The classic HPV-negative pathway begins with polycyclic aromatic hydrocarbons. These are highly toxic chemicals delivered directly to the tissue through combustible tobacco and heavy alcohol consumption. Environmental risks also extend beyond cigarettes. In certain regions, particularly in Southeast Asia and the Pacific, high rates of oral cavity cancer are tightly linked to cultural habits, like chewing areca nut and betel quid. When these carcinogens enter a healthy cell, they cause direct physical damage, snapping the DNA strands. Over years, this relentless damage creates genetic mutations, deleting tumor suppressor genes like TP53, which act as the cell's emergency brakes to stop uncontrolled growth. HPV-negative HNSCC is a disease of attrition. It is the result of a slow, brute force breakdown of a cell's natural defenses over decades of constant toxic exposure. The rising epidemic follows a different biological script. Here, the culprit is the human papillomavirus, specifically the high-risk strain HPV-16, which is transmitted to the throat. The virus slips into deep tonsil crypts, reaching vulnerable basal stem cells. It forces the cell to produce viral proteins E6 and E7. E6 assassinates the cell's P53 alarm, while E7 destroys the RB regulatory protein, dismantling cell cycle checkpoints from the inside. This distinct mechanism leaves behind visual evidence. This is a tissue sample stained for a surrogate marker called P16-INK4A. The diffuse brown staining pattern you see here is the primary diagnostic proof of an HPV-positive tumor. HPV-positive HNSCC is a targeted hijacking of a healthy cell's machinery. That underlying mechanism makes it biologically distinct from smoking-induced cancer. Regardless of how the cancer begins, both types employ the same strategies to survive inside the host. They build a highly fortified tumor microenvironment. To thrive, the growing mass alters its surroundings. It exhausts the local oxygen supply, creating a state of severe hypoxia, and undergoes a mesenchymal transition that allows the cancer cells to detach and spread to other parts of the body. Then, the tumor deploys its ultimate defense mechanism. To avoid being destroyed by the immune system, the cancer cells display a surface receptor called PDL1. When an attacking T cell makes contact with this receptor, it is immediately blinded and deactivated. Effectively treating head and neck cancer requires interventions that can dismantle these fortified immunosuppressive shields while simultaneously attacking the tumor cells. Because these two diseases operate differently, they respond differently in the clinic. Patients with HPV-positive cancers respond significantly better to radiation and chemotherapy, leading to much higher overall survival rates than patients with HPV-negative tumors. That distinct survival gap forced the global medical community to adapt. In 2017, the primary cancer staging system was officially split into two separate tracks to accurately account for the viral variant. Treatment has also evolved to meet the tumor on its own battlefield. Doctors now utilize checkpoint inhibitors designed to strip away the tumor's PDL1 shields, allowing the patient's own immune system to reactivate and fight back. The ultimate preventative weapon against the viral epidemic is the widespread use of FDA-approved HPV vaccines. These can stop the initial infection before the virus ever reaches the tonsils. Understanding head and neck squamous cell carcinoma requires looking past the top-line diagnosis. Acknowledging the dual nature of HNSCC enables doctors to deploy targeted, personalized medicine and effectively halt the rising epidemic of tomorrow.